the mechanism by which these receptors bind their neurotransmitter or hormone in question, and how they actually bring about a response on the tissue that they're specifically located in. We'll start with alpha-1 receptors, right? So this is specific to the alpha-1 receptor. And the alpha-1 receptors behave somewhat differently than alpha-2 or any of the beta receptors. So we're going to consider them first and separate. So norepinephrine will bind to this alpha-1 receptor, right? So here's alpha-1. We have it coupled to a G protein. And as we discussed previously, this G protein has several different subunits. The alpha subunit specifically will dissociate off, float downstream in the membrane, bind to this amplifier enzyme. This is called phospholipase C, right? This enzyme that's downstream in the membrane. And when the alpha subunit binds to phospholipase C, that will activate phospholipase C such that phospholipase C will cleave PIP2. And PIP2 stands for phosphatidylinositol bisphosphate, right? Or PIP2 for short. And so phospholipase C and lipase here, the suffix in this word means that it's going to cleave or cut something, right? When you see that enzymatic term in the name, it means it's going to catalyze a reaction to cut or cleave. So that is exactly what's going to happen. It's going to cleave PIP2 into two smaller subunits, which will act as second messengers. So that will be IP3 and DAG. IP3 being, I think it's inositol triphosphate, and then DAG being diacylglycerol. You don't have to know the long form of these names. You can expect the shortened form on exam questions. So no need to necessarily remember or memorize the longer form, but definitely be familiar with what they are, just in case it comes up anywhere else. But these two second messengers, IP3 and DAG, will then enter the cell, activate other enzymes in the cell, protein kinase C being an example of that. And then protein kinase C will phosphorylate some protein in the cell, and that will generate our cellular response. The other thing is that DAG can also result in calcium release. So this is the endoplasmic reticulum. This is an organelle inside the cell, and its main role is to store calcium. So when the endoplasmic reticulum releases this calcium, that can also be another way that we can result in cellular response. And you don't need to know exactly what the cellular response is. Again, this will differ depending on exactly where we have these alpha-1 receptors, but definitely know the second messenger that will eventually result in that cellular response. So you do need to know that this is a stimulatory G protein, right? The small s here means that this is going to stimulate the amplifier enzyme, activate this enzyme to cleave this PIP2 to make these two second messenger, and then we result in cellular response when these second messenger move into the cell. All right. So that is, we've got an actual schematic of that. So let's kind of go over that one more time. So norepinephrine binds. Here we go. Norepinephrine binds. Here's our alpha-1 receptor. This is a G stimulatory protein, so it's going to encourage alpha-1 to activate phospholipase C, right? Phospholipase C is going to cleave PIP2 into two smaller components, IP3 and DAG. IP3 and DAG can both enter the cell. IP3 can result in calcium release, which can result in our response that way. DAG can result in phosphorylation of proteins in the cell, which can also be another way that will result in cellular response. All right, so this is the case for our alpha-1 receptors. Let's talk about our beta receptors and alpha-2. So because alpha-1 receptors behave so unique in the amplifier enzyme that they use, in the type of second messenger that they create, we're going to consider those separately. Alpha-2 receptors and all three beta receptors, which is what we're considering here, all three beta receptors and alpha-2, they use the same amplifier enzyme, which is adenylate cyclase, not phospholipase C, and they are also going to generate the same second messenger, which is cyclic AMP, not IP3 and DAG. So we can kind of consider alpha-2 receptors and all three beta receptors in the same general scenario, although they're going to be located on different tissues. So while we're thinking about them in the same general scenario, just because they have the same amplifier enzyme and the same second messenger, still understand they're going to have different effects depending on exactly what tissue they're located on. So for example, beta-3 receptors are only found in adipose. So although it's the same second messenger, that second messenger is going to bring about a different response in adipose as, for example, beta-2, which is found in the lungs. All right, so let's look at this. The other exception I want to point out here is that although, again, we're looking at the same amplifier enzyme, same second messenger, beta receptors are going to activate that, right? They have a stimulatory G protein, so they are going to activate this process. Alpha-2 receptors have an inhibitory G protein, so they are going to block this process, right? They're going to inhibit this process, and that is still going to result in the cellular response. So let's look at that sequence of events. Let's consider the alpha-2 side first. So norepinephrine or epinephrine will bind to our alpha-2 receptor. Because this is coupled to a G inhibitory protein, when the alpha subunit dissociates off, it's actually going to inhibit the second messenger, right? So we're going to block the production of second messenger. And because we have blocked adenylate cyclase from generating cyclic AMP, that is also going to result in a cellular response. I want to make it clear that even though we're blocking these events, the blocking of adenylate cyclase and the decrease in cyclic AMP is what results in the response that happens on alpha-2 receptors. All right, let's consider the beta side of things. And again, we're looking at all three beta receptors here, although we're only illustrating it on one membrane. We want to think about all three of the locations that beta receptors are found. So again, we have a G stimulatory protein. In this case, the alpha subunit dissociates off. Because it was a stimulatory protein, it's going to activate or increase the amount of cyclic AMP that is made by the amplifier enzyme, which is adenylate cyclase in this case. So we generate cyclic AMP, and that cyclic AMP will stimulate protein kinase, which will phosphorylate some protein in this cell, again, depending on where we are specifically, and that will generate our cellular response. So while we have described the same amplifier enzyme, the same cyclic AMP, there are very different things happening with regard to these two, the amplifier enzyme and the second messenger. And again, I want to reiterate that although we are blocking or inhibiting the production of second messenger when we have alpha-2 receptors being bound by norepinephrine, that is still going to trigger or result in the specific cellular response that is acquired on the tissues that have alpha-2 receptors. Okay, so the blocking is going to generate the cellular response on alpha-2 tissues. Okay, the other piece that I want to distinguish here, and again, this is where this becomes a little bit of a confusing concept, is that we have beta-1 receptors, right? We have beta-1, beta-2, beta-3 on this side. Beta-1 and beta-3 are going to be excitatory effects on their tissue. Beta-2 is going to be inhibitory in its effect on the tissue. So when we don't want to confuse what's happening with the second messenger Whether we are increasing or decreasing second messenger, we don't want to confuse that with what's happening on the tissue. Those are two separate responses. The effect to the second messenger versus the effect to the tissue. So those are two separate things. If we think about alpha-2 receptors, these are going to be excitatory to their tissue, right? In that they're going to contract smooth muscle on vascular, on blood vessels, right? But in order to do that, it's going to require an inhibitory G protein to block cyclic AMP to result in that excitation on smooth muscle. If we consider beta-2, for example, beta-2 is going to have a G stimulatory protein. It's going to result in increase in cyclic AMP, but that cyclic AMP will result in an inhibitory response, inhibitory response on the specific tissue in question, which happens to be the lungs. Okay, so we definitely don't want to mix what's happening with the second messenger with the type of a response or effect that we see on the tissue. All right. So here is a really great summary table to just summarize this. This is something that I'm going to encourage you to commit to memory. You'll see this come up in a lot of different forms in questions. So know the effector organ that we find these receptors on, know the differing affinities, right? So alpha receptors are always going to prefer norepinephrine. Beta receptors are going to differ in their affinities. Know the signal transduction mechanism. In other words, what do they do? What second messenger do they regulate, and what do they do to that second messenger, whether they inhibit it or activate it? Also know the effect that they have on the tissue in question, the effector organ or effector tissue in question. All right, and here's where I'm going to reiterate that we can have a receptor that results in inhibiting cyclic AMP, and that can still result in an excitatory response on the tissue. We can also have a receptor that results in activating its second messenger, which will result in inhibiting its tissue response. So definitely think about these two things as separate, but also, know which ones are going to be increased versus decreased. Another really nice summary table here. This one, not so important for you to commit to memory, but this will be nice if you're interested in looking at some of the medications and drugs and pharmacologic components, their mechanism of action, and then how that brings about a different tissue response. So this is how we can use our knowledge of these receptors, and we can actually manipulate these receptors to bring about some desired tissue effects. So a good example of this is if we look at isoproterenol. So isoproterenol is a beta agonist. And when we think about the word agonist, it's going to mimic the response of norepinephrine binding to a beta-1 or beta-2 receptor. What this is going to do is, if we think about beta-2 specifically, beta-2 receptors result in dilation, bronchodilation. And this is what we mean by the effect on the tissue. So although when norepinephrine binds to a beta-2 receptor, that results in increase in cyclic AMP, if we look at the tissue in question, the result is to inhibit that tissue and result in opening up of the bronchioles, which is dilation, and that can be considered as an inhibitory response on the tissue in question. But that is the desired effect, right? That is the desired effect for the sympathetic nervous system. And if, again, we step away and kind of zoom out, if I'm fighting or fleeing, if I'm exhibiting that fight or flight response, I would like my bronchioles to be opened up. I would like for the lumen to be larger so that I can take in more oxygen, so that I can increase my oxygen supply to my skeletal muscles, to my other organs, so that I can run, right? So that I can have more oxygen being supplied for energy, right? So that I can be that can make movements to get away from that threat. So this is the desired response that is going to be in alignment with the sympathetic nervous system and the sympathetic response. And so we can use that knowledge. We can agonize these receptors, and this can be beneficial for asthmatic patients. If they have narrowing of the bronchioles because of airway irritants, or just because they have whatever pollutants in the air that may be causing constriction of the airway, we can use this knowledge, mimic the action of norepinephrine binding to this receptor, and we can have the airway dilating. So this is how we can, again, use our knowledge of these receptors and actually mimic some of these responses and have some therapeutic effects by manipulating these receptors. And so that's an example of an agonist. If we think about an antagonist, right, something that's going to block that response, we can look at, for example, glaucoma. So if we block beta-1 receptors, that is going to block the sympathetic response, and that is going to encourage the parasympathetic response. And so that's going to somewhat result in decreasing the intraocular pressure. And so we can begin to, again, manipulate that understanding and mimic binding or blocking of these receptors to bring about some desired effect. All right. Let's go on to looking at the actual neuroeffector junction. So the neuroeffector junction is the synapse between our postganglionic neuron and our effector organ or effector tissue. So here's our postganglionic neuron. And if you remember correctly, we had two neurons in series, right? We had a preganglionic neuron, and then we had our postganglionic neuron. So the junction between the postganglionic neuron and the tissue in question is going to look very different than the junction between the preganglionic neuron, right, and the synapse, right? This is going to be that ganglia. So this junction is going to be a regular synapse. It's going to be a regular neuron to neuron synapse, but the neuroeffector junction is going to be a little bit different. It's going to be the junction between a neuron and some other type of tissue. This can be cardiac muscle, right? This can be smooth muscle. This can be glands, right? Or even adipose. So the junction here is not going to be a neuron to neuron synapse the way that we have described previously. It's going to be a neuron to some other type of tissue. The other difference between this junction and a traditional synapse is that we're going to have varicosities. So these varicosities are these swellings along the end of the neuron. So whereas in a typical synapse, we have the neuron ending in an axon terminal that looks something like this, here we have a neuron ending in several different swellings that will actually have the neurotransmitter being stored in vesicles within the varicosities. So if we look a little bit closer at these swellings, we can actually see the synaptic vesicles inside of the swellings, and it's going to contain the neurotransmitter that's being released. And it's going to behave just like a postganglionic neuron, just like an axon terminal, in that the calcium channels are going to be activated here when the action potential arrives at the varicosity. Calcium is going to rush into the varicosity or the swelling here. That's going to trigger the release of the neurotransmitter, and it's going to cross the junction, bind to the tissue, and then result in the response. So the mechanism of transference, right, the mechanism of exocytosis, of neurotransmitter release is the same. It's just that the anatomy here is a little bit different, and the distance is also somewhat different. So it's a larger gap between the varicosity and the tissue than, say, a preganglionic neuron and a postganglionic neuron. Okay. All right. I'm not going to go any further. We'll pick this up. In fact, when we meet on, or the content that I release for Wednesday, I'm going to pick up with this. We're going to finish up this, and then it's going to get into the somatic nervous system.